Hello, everyone. Yes, thank you. So welcome to everyone to the biodiversity seminar. It's a pleasure to me to introduce Iva Franić from Zagreb, Croatia. Iva made her Master of Science in Ecology and Nature Preservation in the University of Zagreb. And I was trying to remember when I met first time Iva, and I guess it's around there 2016 or 17, when Iva came for her PhD to WSL. Her PhD was a collaboration work between Simone Prospero from WSL and René Eschen from Gabi in Delemont. Her PhD was entitled Global Assessment of Tree Pests and Pathogens for Improved Biosecurity. For her PhD, she studied insect and fungal communities of tree seeds and twigs to determine the main drivers of the observed diversity patterns and to assess the phytosanitary risk associated with the movement of plant material. And I think this is really a key point in your career, trade and seed movement. Yep. She continued her research after the PhD. She continued her research as a Swiss National Foundation postdoctoral fellow in Southern Swedish Forest Research Center at the Swedish University of Agricultural Science in Alnarp. Is that right? Okay. Where she continues studying seed microbiota, the diversity of microbiota, transmission to the seedlings, phytosanitary treatments of seeds, and utilization of various molecular methods for the assessment of fungal communities in plant tissues. Since September 2023, Iva is employed as a tenure-track scientist in phytopathology group at WSL, where she continues working on tree microbiomes. The diversity patterns of microbiomes, assemblage processes and functions, we always with an overall aim to reduce the risk of introducing novel pathogens through plant trade and to identify potential beneficial microorganisms that could be used to increase trees' resistance to biotic and abiotic stresses. Iva, I'm looking forward to hearing your presentation.
Thank you, Carolina. This was very exhaustive introduction, but I think I don't need to add more. You know approximately where we are working today, and I'm focusing today mostly on the seed story and microbiomes in seeds, and that's because I've done that from my PhD and then continued in the postdoc, although I worked also on different parts of trees. But as Carolina said, everything, it's what I'm combining in my research is this biodiversity research, so looking into biodiversity patterns, drivers of the biodiversity, but from the phytosanitary perspective. So what does that mean from the point of introducing new pests and pathogens to the new areas? So last week or 2 weeks ago, Irene placed you in soil and she did a really good job with picturing how it looks like in the soil and what does it mean to be a microorganism or an organism in the soil. Today, I would like to place you into the seeds. So many of you know, or all of you know, that seeds play a crucial role in tree dispersal. They play a crucial role in sexual reproduction of trees, and they also offer protection and food supply to tree embryos. So they're quite important in the life cycle of trees. Here on the picture, you can see that they appear in different shapes and forms, mostly depending on the dispersal strategy, but what we all know, so conifer seeds come mostly in cones, then very well known, I can also use this, not that one, this one. samaras, of maples, and different nuts are just some different types of seeds. So besides these morphological differences in how seeds look like, they also differ in their physiology. Most of them have very high levels of defense chemicals, but also differs depending on a species. And they go through different physiological changes throughout their life cycle or throughout the season, where they are characterized with different levels of desiccation, so levels of moisture, and also how deep they enter dormancy, which means how they stop their metabolism during unfavorable time, which is for most temperate species winter. For these periods of time, they are representing a quite low nutrient harsh environment that is not great for many organisms. However, some of the organisms are quite fine with it. And one group of those organisms are, for example, fungi. So here you can see this is the acorn, and here are the microscope pictures from the acorn parts. The green parts of the pictures are fungi. And what you can see is that they really appear outside of the seeds, on the seed coat, but also in embryo and inside of the seed. So all parts of seeds, fungi can live in them. This is also the case for bacteria. So here also microscopic pictures of bacteria inside of the tissue, as well as fungi all over. So how do these microorganisms get into the seeds? There are different ways of transmission, but here, if you look into the seed, you can see that they can come from the reproductive parts, so through gametes, pollen, or ovule, and they can also come from a mother plant asexually, so not from the sexual cells. And then when the seed is formed, it consists of this community of microorganisms that come from the mother plant. And then in the nature, they normally overwinter or are dormant, either in soil, litter, or sometimes on the trees, but rarely. And during that time, they acquire new microorganisms through interactions with the environment. So from basically horizontally transmitted from the environment. And in this stage, the survival of these communities and the harboring of new ones depends on different conditions, for example, treatment, storage, and production in the production practices. After the dormancy period, so during dormant period, many of these organisms are inside on a very low metabolic level, also some even in form of spores, so very inactive. But once the germination starts and metabolic processes are triggered, there is a lot of simple sugars that are going around, and these communities can flourish. And then what happens is that they are transferring to different parts of seedlings, so they can go to the roots, to the stems and leaves, and they're also getting new portion or horizontally transmitted microorganisms in the plant with which they are interacting. So this is somehow what is also important for what I will talk about later, but like how these communities are assembled, basically. If we look into diversity of these communities, we are mostly talking about bacteria and fungi. So you can see here that bacterial richness is quite higher than fungal richness in seeds, and they differ in the ratio, so which is better to look at here. So you see that some trees, some species, plant species are dominated by prokaryotic diversity, and some by fungi. I don't think you see it here, but if you, or you see, but there are many names. These are mostly grasses and vegetables. The only tree species that is here is this oak. So that's one study that is represented in the quite recent meta study of seed microbiomes. So based on this, my point is just that most of it really doesn't come from forestry, right? Which is a pity, because all these studies do show that these organisms can have really important impact on the seedlings and on plants later. They influence or they provide benefits by optimizing nutrient uptake. They can act as antagonists on competitors and thus protect the plant from other pathogens or insects. They can improve quality of soil and water, stimulate plant metabolism, and increase tolerance to abiotic, but as I said, also biotic stresses to the plants. However, seeds can also transmit plant pathogens. So many of these organisms that are inside of the seeds are, as I said, dormant. They're mostly doing nothing. However, later, in the later stages of plant development, they can become pathogenic. And some examples here are in this, on this slide, I focus now on trees, although there is not much done on trees. What is done is that we are aware of certain plant pathogens which are seed transmitted. And these are mostly here examples of pine pathogens and on the other side of eucalypt pathogens, which also indicates that the research is strongly biased towards economically important plantation tree species and pathogenic fungi of those. So we do know that nowadays, forestry depends on a constant supply of good quality seeds. This is because lots of 1/3 of European forests are, for example, artificially regenerated. And while the propagation material for artificial forest regeneration is often locally sourced, we still know that around 25 million kilos of tree seeds are traded internationally every year. What we expect is that these numbers might even increase, and this might be because of different reasons. One of them being if we ever really start seriously doing assisted migration for climate change, we will need to think about how we will move those plants, and that will probably be seeds. And the other reason is that many countries are not being able to rely on their seed supplies due to unexpected environmental disturbances, such as fires or just tree dieback. So they need to import seeds from other countries in order to be able to grow their trees. So if we look into this international movement of tree seeds, what we can see is that the numbers are stable over time, more or less. So the amounts are quite high. And what we can also see is that the patterns are quite hectic and unpredictable. But what is symptomatic is that here you can see in the first five-year period is that colors are not crossing much. So most North American seeds are imported in North America, most European into Europe. As we are moving later, we can see that this crossing of colors is going up and down. So colors are mixing, which indicates that there are significant movements of even intercontinental movements of seeds. And this all becomes quite problematic because we know these plant pathogens, when they are moved, they're especially dangerous if they are moved from non-native to non-native areas where they encounter either naive hosts or they are not having natural enemies. So this kind of plant movement is what we worry about. And on top of that, there are only few restrictions that target tree seed trade in Europe. So in Europe, since 2019, all tree seeds need to have phytosanitary certification, which is not a real phytosanitary measure. Phytosanitary certification is basically a declaration of a seed lot. It tells you what it is and where it comes from. It tells you that it's safe of pests, but there are no requirements for checking these seed lots before to say that they are safe of pests. So basically these assumptions are done based on visual checking normally. And the only specific phytosanitary requirements are in place for pine, pinus species and pseudotsuga, so Douglas fir species. And these, because they are likely to carry Fusarium cincinatum, which is the only quarantine forest pest that is known and seed transmitted. For those of you who are not from biosecurity background, quarantine forest pests in this case are the ones that are absent from Europe, and we want to keep them away, right? So because all of this, so this high seed trade plus few restrictions, the first study on seeds that I've done was looking into various tree species belonging to angiosperms and gymnosperms, coming from 3 continents, so North America, Europe, and Asia. And what we did is that we ordered seeds from commercial seed suppliers. And then we used those seeds to assess fungi and insects in them. I'm talking about fungi today only. So basically we took 100 seeds per seed lot and used half of it for plating. So we grew fungi on plates and identified them by Sanger sequencing. And we used the other half of the same seeds and pulled them per seed lot and assessed fungi by using DNA metabarcoding. And what we see, or what we revealed by this study, is that the infection rates of these seedlots were super high. So we isolated or sequenced fungi from all seedlots. The diversity was also very high. We are talking about almost 2,000 fungal taxa. So I'm mixing in this presentation ESVs, OTUs, depending on which bioinformatic pipeline we use, but I will just refer to them as fungal taxa. And they belong to around 200 genera and species. What is very typical for these data sets is that not much can be identified. So we have around 60 and 30% that are assigned to genus and species. And when we look into what they are around 30% are plant pathogens, which is quite high. We look into diversity patterns, basically taxon richness. What we can see here is that on every continent, angiosperms have higher diversity than gymnosperms, and overall Asia has higher diversity than the others. But what is more interesting in this case is to look into these communities. So basically what you can see on this graph, or on this figure, is that shapes that are closer are more similar. The ones that are further apart are less similar. And then you can also see the splits in communities. So first axis separates gymnosperms from angiosperms. And indeed, only 19% of fungal taxa are shared between groups. And then the next separation is between Europe, North America, and Asia on the third level. And again, 65% of fungal taxa is unique for one of the continents. Interestingly, in this case, Europe and North America share 3 times more fungal taxa than each of them with Asia, which is interesting but not surprising, because historically Europe has traded much more with North America than with Asia. which means that we exchange a lot as well. What is a bit worrying is that the imports from Asia are increasing, and at the same time we show that they have very unique fungal communities with many, with very high richness. So that is all yet to come. And the last thing that we did in this study is that we did short pest risk assessment, or in this case, pathogen risk assessment. So we looked into all these identified plant pathogens and then looked into if there are any that are not yet recorded in Europe and that are known to be pathogens of woody plants. And we have these, I think there were 11 species. So basically we didn't go very closely into it later, but for us was important to show that there is evidence that potentially dangerous pathogens are being moved. So because in this study we face this problem with identification of fungi, what we decided to do next, or what was basically done next, and now it's mainly led by Jana here, is that we tried long-read nanopore sequencing for identifying these pathogens to see if by sequencing longer reads, we will be able to get also more accurate identification, because Illumina sequences that we are using are very short and are limiting the ID. So basically, the same samples were used for nanopore sequencing, and then we compared it with 2 primer sets that from Illumina, right? So 2 primer sets from Illumina plus nanopore. And what we can see is that on top, these numbers of the first figure show that a different number of OTUs was revealed from each method. You can ask later why or how, but number of genera was basically similar. So, and then if you look into the other one, you can see that also the composition of genera was very similar. So basically, these different molecular methods reveal very similar fungal taxa, which is good news for us, because that means that all these studies are quite comparable, that use different methods. However, it's a bad news because we didn't get a better ID. So, and the reason why we didn't get it can be different. One might be a high error rate of the nanopore back at that time when we did that. Now it's a bit better, or a lot better. The other reason might be that although we are sequencing a long or whole ITS region, reference databases are still based on ITS 1 or 2, so we are basically limited by what is sequenced out there. Okay. Another methodological study that is not on the same samples but similar is, so besides getting a good ID, what we also need to know is, are the organisms that we are detecting alive? And the problem with DNA is that DNA can be detected when organisms are dead. So what we decided to do, and this was part of my postdoc in Sweden, we used RNA metabarcoding for looking into viable communities in these seeds. So the method is the same, it's very similar to DNA-based metabarcoding, except we extract RNA and then synthesize cDNA. And then, but the rest is pretty much similar. So we did this with 3 tree species, 5 seedlots each, and looked into plating DNA and RNA. I'm presenting only DNA and RNA here, with an idea that DNA would reveal this very diverse community, and then RNA would reveal live portion of it, while culturing, because it's much less sensitive and much more biased to fast-growing fungi and ones that are abundant, would reveal just a small portion of this viable part. And this is what we get. So we get more reads in RNA data set, more OTUs in RNA data set, more genera in RNA dataset. And this is the case again for all 3 species. So just the diversity in RNA dataset is wild. And then if we look into the communities, we also see this clear separation between DNA and RNA communities. And methods indeed explain around 30% of variation and species around 13. However, this is based on presence-absence data. As soon as we look into dominant communities, methods explain only 4%. And then you can see the overlap here. As you go to more dominant communities, overlap is more and more, and uniques are less and less. There is, I'm not sure why this is happening. One of the possibilities is that it's a technical artifact. Another possibility is that, as I mentioned previously, that seeds, because they are dormant, they're also dominated by dormant stages of fungi. So if very abundant fungi are in spore, present there as spores, they will be detected only by DNA, but not RNA. And it's possible that there are just this huge amount of spore DNA is masking the diversity of rare species that is then revealed by RNA. But that's probably something to explore more in future. After looking into diversity and finding the most optimal method to assess diversity patterns, identify species, and find viable communities, I was also interested in looking at diversity patterns and their drivers. So what is driving, what are the main drivers of differences? So we did a study in which we used the same 3 species as in the first study. And in this case, we sampled it in botanical gardens in Europe and North America reciprocally. So it's like a common garden experiment. And then we looked into relative importance of host identity, different site-specific variables, and distributional range, so native versus non-native. Again, here using DNA metabarcoding and 100 seeds per seedlot. And what we have found is that host identity is the key driver of community composition of tree seed-associated fungi. This is the case when we look into presence, absence, and when we look into dominant communities. Here on this heat map, you can see a bit like I call core communities. So there are no fungi that appear everywhere, and there are no rare fungi, only for the illustration how the separation exists. So here you can see samples that are coded by host genus and continent, and fungi here. Basically what you can see is first separation in clustering is between gymnosperms and angiosperms. Next one is between larix and other conifers, and fagus and acer, and so on and so on. So basically separation happens angiogymno, then genera, and then later species, and only somewhere there continent appears as important. So much higher importance of host identity. So because of this high importance of host identity in shaping these communities, we were wondering if this maternal inheritance might be quite high. So because basically if seeds were collected in Europe and brought to North America and they are still not much different there, is it possible that they are just carrying a lot of these microorganisms from generation to generation? And this was, well, when I started doing it, for me, it was quite controversial thought, because by then, no one really, it was not known that this vertical transmission exists in trees, or it was not that common. In comparison with grasses, for example, when there are endophytes that just are transmitted generationally. And the reasons for this is because seeds develop much longer, chemistry is much more complicated, structure of organ is complicated, there is a lot of things coming from outside. So it was really, they are not assumed to be vertically transmitted. Another reason is that most studies focused really on looking into if certain pathogens can be vertically transmitted. And that's a bit of a problem because pathogen abundances in seeds and frequency in seeds are very rare normally. So if you collect seeds from outside and then you grow them, plus inoculation levels that are low. So that was, not very often that they can be vertically transmitted. So I decided to do also, this was in Sweden, a very simple study, just to surface sterilize seeds and grow them in sterile conditions and see what is going up in the seedlings. So there was a lot of trial and error. I did it in these tubes on artificial media, then in semi-sterile by sterilizing soil, but not being able to exclude everything from outside. This can be done much smoother. But in any way, what we found is that 70% of OTUs that were found in seeds were also found in seedlings. So this is around 70% in fagus, around 84% in pine, which was quite surprising. And then when we look into what these fungi were, we could see that again, similar patterns as before. Many of them are not identified, around 30% are pathogens. And all of this now is opening a whole new door to start exploring really how much of these pathogens go up in the seedlings, but also how do they persist in time? Because it might be that amounts of these fungi that are being transmitted are very low, and that what's happening is that as soon as you put the plant out, they will be overcompeted with whatever is coming from outside. In any case, this is the short overview of my seed studies. And I would just like to summarize quickly. So basically what I wanted to say today, and I hope I did, is that the diversity of tree seed fungi and their function especially, but also bacteria, not only fungi, is still largely unknown. However, we do know that high volumes of seeds are being moved across countries and continents with these unpredictable and variable patterns over time. And based on the studies that I've done, we know that these seeds harbor taxonomically and functionally diverse fungal communities, that they include potential pathogens that can be transmitted to the seedlings, which indicates that the likelihood of risk of transport and introduction and establishment in the new environment is high. But before making conclusions and putting bans on seed movement, what we really need to know is much, much more. First is improving the ID of these species, mainly by finding methods that will allow us more precision, but also populating databases, because without reference sequencing, we are going nowhere. We need to know more about viability, explore not only RNA, but different methods to see how viable these communities are, or how being able basically to distinguish between alive and dead. What I mentioned in the previous slide, it's very interesting to look into persistence in seedlings over time, so of microorganisms, so basically to look into temporal variations, which there is not a lot done there, because we all know that it takes time. And then of course, function, reproduction, and like very, very basic biological information that we are missing before we can really make conclusions about what's going on. And that's it from me. Thank you for your attention. Yeah. You can ask me questions.
So thank you very much, Iva, for this great overview of your work. We open now the microphone for questions. Yes, wait, because
Thanks, Iva. I like it a lot. I'm just thinking
Now
about...
a critical question.
No critical, never. No, just ignore it if the question doesn't make sense.
I will not ignore it.
Maybe I didn't get it, but I'm wondering about that DNA, RNA thing and so on. How did you assess the diversity? Did you assess it externally by checking it against the external database, or did you also check it within? So DNA dataset against RNA dataset by like reciprocal blast or...
So basically, yeah, DNA and RNA dataset are bioinformatically processed together. So we take reads from DNA and RNA, pull them, and then process them.
Mm-hmm.
So, yeah, no. Or you process them until one point separately, but then you merge the sequences.
And they were matched again.
Yeah, they are matched at the same time.
Okay.
So it's no bias from processing them separately. But just one more thing, I did it both. So, because in the beginning I had DNA and RNA that were processed separately and I was in a rush to write a report, so I used it like that. Then I started thinking, oh, maybe it's because of that. So then we did it together and it's the same. It's the same patterns that are, yeah.
okay, thank you.
Well, thank you very much. It was very interesting. I'm just wondering, you are saying that you have around 30% of pathogens in the seeds, but do you know how many of these pathogens are specific to the host?
Yeah.
Because it can be very broad pathogens that are not related to, yeah.
So, yeah, I was maybe not totally precise. I'm trying to use term potentially pathogenic, potentially plant pathogenic genera, because they are not pathogens, right? They are doing nothing in that seed because seed is asymptomatic. They might never do anything. And part of that is because they're having these non-specific host pathogen associations. However, we do in this study with the heat map, we also did the indicator species analysis with looking into which pathogens appear always or more significantly in certain genera than others. So in that case, we can talk about specificity a bit more. Again, that specificity of where they appear, not of where they cause a disease. That's the next bottleneck, and that's probably why most of them are not dangerous. However, what is problematic is even if we introduce host pathogen association that is not pathogenic, right? In the new environment, you are having new pathogens that might jump to new species if there is a host. But yeah. Is that an answer? Oh, it was good, okay.
Other questions? Yes, no?
Thank you, Iva. I have a question a bit more from the very beginning of your presentation about this idea of how the microbes are inherited. And I'm wondering, do you know, so I understand how microbes can be inherited from the leaves area and the above ground area. Is it possible to inherit microbes from the roots? Like the root microbiome can be inherited somehow? Do you know anything about that, or is completely out of your scope?
Yeah. Well, what I understood, I mean, this is very like theoretical framework, right? But what seems to be happening is that, so by sequencing different tree parts, we see that there are differences in communities depending on organs. However, we find things that you would not expect there. So there is diversity is huge. I suspect things are also going up and down quite a lot. So in that sense, whatever is in the roots can also get in the seed if it's special, if it's adapted to move, right? Are you asking about this if am I understanding well?
Yes, yes.
Can
So
root
I'm just thinking,
pathogen be moved to the seed and then moved transmitted to next generation?
exactly. So
Yeah.
yeah, I mean, not only pathogens, I'm also thinking of mycorrhiza or things like that. If they will be able to be transmitted by the seed, I mean, it's not.
So I know that arbuscular mycorrhiza are normally not transmitted by the seed, which is also something, I didn't show it here, but something that we found in this seed to seedling transmission. So I found a huge number of reeds from glomus in both seeds and seedlings. Okay. So in that sense, I don't know. Yeah, I need to talk to someone who knows more about if that's remotely possible. But other mycorrhiza are known sometimes to be also seed transmitted. So it should be possible. Yeah.
Cool. Thank you.
Yeah. Because that's also, sorry, the last picture that on this one is that what is in the seed also goes up and down. So I guess it's possible also to shrink it and move it back.
Okay. Thank you very much also. Maybe connected to that, in case you even know, do you know if these fungi that you have in the seeds, it's their kind of main lifestyle to be vertically transmitted, or is it kind of a drive-by so they can be horizontally and vertically transmitted? Do you have any examples that you already know?
So that's also, thank you. Yeah. It's similar to what I was mentioning before about this. So now I will forget, but 3 endophytes belong to the type 2 or 3. Yeah. So endophytes that are not known or not believed to ever be vertically transmitted. So these are most of 3 endophytes. And this idea that we are finding things that are going up is quite surprising. However, we are not proving yet that they are going again into the seed and next generation. So we are only proving they are going from seed to seedling. They might disappear at that point and never be transmitted. So really, based on what we know, it seems that 3 fungi are acquired mostly from the environment, so horizontally. And this idea, I mean, we really need to look. From my data, I don't see that there are some specific fungi that are known to only be vertically transmitted. So most of the fungi we found in seeds and seedlings are environmental, like well-known pathogenic or environmental genera. Nothing really, that would say I'm vertically transmitted only.
Yeah, hi. In your C2 seedling experiment, did you see like any effect on the seedling growth performance? Like did the seedlings from the sanitized seeds like grow worse or better?
We didn't measure that. It was a hectic end of the experiment, beginning and end. Yeah, but that's a good point. We are now doing. So Martina is not here, but Martina Hobby, they have this joint Ukrainian Swiss long-term project. So they are supporting a collaboration with Ukrainian researcher who is doing testing of different bacteria and seed coating on plant performances. So she is measuring the growth, different physiological parameters and finding like really cool candidates that could not even that much fight specific pathogens, but really boost seeds or seedlings to grow and in that sense combat antagonists. Yeah.
Other questions? Oh, okay. More questions? No? In chat? Okay. So if no one is asking, I have a question, but it's more just an idea, not a question.
Yeah.
So we are talking about endophytes, and I was just thinking if we have to rethink our concept of endophytes in the way that endophytes are able to infect plant tissue and to maintain their existence within this plant tissue. And then only because we don't see in our human life, we don't see maybe a disease, it doesn't mean they are not really pathogenic. And maybe we have to rethink about it, about this concept of endophyte, that only symptoms count. And
I guess we will also need to move from like classical taxonomy that we are doing for now and just assuming function based on taxonomy, right? Because in that case, potentially pathogenic, what does it mean? Again, this host association, environmental factors, will it ever be triggered or it will not be triggered? Yeah, it's definitely, I will need a new layer of specificity in thinking about these things.
Thank you very much. Very interesting topic and going into philosophical ideas. So if there are no more questions, I thank you very much, Iva, for this very interesting presentation.
Thank you.